All right, guys, we are going to continue in our notes. We're going to talk about something called mutations. This is topic 6.7. And I know when I say mutations, probably some of you all instantly flash to mutants, right? X-Men, mutations, mutants, which is kind of interesting, honestly, that we think that mutations are this significant, this severe, and that can change us that much. And I hope that one of the things that we can see by the end of this is that some mutations are pretty impactful and other mutations do absolutely nothing to us. So let's start first with what are they? So mutations are just alterations in an original DNA sequence. And these alterations exist for a couple of reasons. So up here, we see several different environmental impacts that could potentially alter, change our DNA. Everything from environmental radiation to chemicals that we might expose our bodies to, even to infectious pathogens like viruses and bacteria. So lots and lots of things could possibly alter our DNA. Everything you see here in this diagram would be considered a mutagen. And so mutations are caused by these environmental exposures, these mutagens, or they could, I should say, or they could be caused by mistakes in the process of DNA replication. So errors that DNA poly 3 did not pick up on. There's a quick definition for you of mutagen. Again, these environmental chemical exposures that we see in that top diagram. So what are mutations, alterations in an original DNA sequence, and why might we get them? Because we've been exposed to something or because our DNA replication process had an error that went unfixed. Now, last thing here on this slide is kind of what I was talking about in the beginning. Mutations are given a little bit of a bad rep. Although there are mutations that are harmful, you will see throughout this section of notes that there are mutations actually that are not bad. There are mutations that do nothing, right? They have no effect on us. And then there are mutations that can actually be beneficial. So if we're talking about a beneficial mutation, we're talking about something that actually helps an organism to survive. And a great example of that in the human population is something that has been recently discovered, I would say probably within the last decade. We have discovered that some people have a mutation in a receptor on the surface of some cells in their body, and that receptor is called the CCR5 receptor. So people that have a CCR5 receptor mutation actually cannot be infected with the virus HIV. And so that's a great example, and you can kind of see in this diagram over here, a cell model where the cell does have that receptor. So these little red squigglies, those are the CCR5 receptors. If you do have CCR5 receptors and you are infected with HIV, the HIV lands on the receptors, and that's how it is brought into the cell, and it starts its replication process in your body. If you don't have those receptors, guess what? Your HIV, even if you're exposed to it, does not infect those cells. So that's a really good example of a mutation in the gene that codes for those CCR5 receptors actually being beneficial because it is protective against this virus. There are a couple of types of mutations. So let's move into the section of our notes that have a couple of charts. So first we'll talk type of mutation, and then we'll talk the effect of that type of mutation. So there's two different broad categories for types of mutation: point mutation and frame shift mutation. So for a point mutation, we're looking at swapping out a nucleotide in the original DNA sequence for a different nucleotide. This is called a substitution. And so really the name point comes from the idea that really there is a single point of impact. There is a single nucleotide that has been swapped out. In this diagram right here, you see an original sequence, and then you see that sequence that has a point mutation. So the original sequence TAACT, here you have TAAC, swap out this T for this C. Now it just exchanges, it swaps those nucleotides, and that may or may not have an effect on the amino acid that gets shuttled into the ribosome. What it doesn't really impact is the sequence of codons, right? So we had 3 codons here that would be read, and then the next 3 codons would be read. Same thing here. By substituting, we still have our same first 3 codons, and then this would be our next set of 3 codons. And so we're not really shifting what is called the reading frame of the messenger RNA. We're still reading it in the same sets of 3 letters, except one of those letters is going to look different because of the point mutation. The other type of mutation, a frame shift mutation, is when we alter the reading frame. And these are the results of inserting or deleting a nucleotide without replacing it. And so an insertion or a deletion is going to shift how we read those codons. It literally shifts what we call the reading frame of the codon, and that's where it gets its name, frame shift mutation. So this diagram down here, if we look at it as a whole, is showing us an original sequence and then an insertion. So we have TAACT, and here we have TAAC. Here's the original T, but we've inserted a C in between. And so that literally pushes all of these nucleotides down, and it shifts the reading frame. So when we start reading our nucleotides in codon sections, we're reading it just a little bit different because we have either added in or we have taken out a nucleotide. So we can talk about mutations as what type of mutation. Is it a point mutation where we just swapped a letter for another letter, or is it a frame shift where we have inserted or deleted a nucleotide? So those are the types. Now let's talk about the effect that those types of mutations might have. And all of these effects could be applied to either a point or a frame shift mutation. So for effects of a mutation, you have something called a nonsense effect. And a nonsense effect occurs when the mutation codes for a premature, an early stop codon. And so the protein actually doesn't get fully built and is not fully functional. So if the mutation causes the protein to stop being produced early, that's a nonsense mutation. And here is a little picture of the representation of that. So here's our DNA sequence. Here's our messenger RNA. If we experience a mutation in any one of these codons that codes for an early stop, we wouldn't get the rest of the amino acid sequence. The next type of effect is called a missense. And a missense means that the mutation that exists codes for a change in the amino acid sequence, which means when we go to read the codon, the mutation changes the amino acid that we're reading on the codon chart, which changes, in most cases, changes the shape of the protein that's being created and affects the function as a result. Right? We know structure determines function. And so if we change the structure of the protein, we're going to change its function. So here's a good example of a missense. So here's the original sequence that codes for amino acids MET, GLU, ALA, and GLI. And here's what that protein looks like. Over here is the mutated sequence. So we just mutated one letter. So this could be a substitution mutation, just one letter, but that change GLU to ASP. So we have MET, ASP, ALA, and GLI. Doesn't look too different, but the protein that is created, the way that it folds up is different, and then it does not function in the same capacity. The last type of mutation is called a silent mutation, which you could probably guess what that means. The mutation actually codes for the same amino acid. And so if you think about your codon chart, there were a couple of codons that all coded for the same amino acid. And so maybe the mutation changed the codon, but the codon still codes for the same amino acid, which means the protein doesn't change. There's no net effect, and that's why this mutation is referred to as a silent mutation. So some mutations are good, some mutations are bad, some mutations have zero effect on us. And so in looking at mutations and their impact, we're now going to do a little bit of practice. And so the paper that I want for you to get for the practice here is the one that has the big muscular cow on the front of it. This is a Belgian blue mound cow. And this cow specifically has a mutation in a protein called, it has a mutation in the gene that codes for the protein called myostatin. And because they have this mutation, they are able to put on incredible muscle mass, and they look like these huge, beefy, bulky cows. And so what you're going to do now is the worksheet that talks about this mutation. So you're going to read a little bit about the protein that's being mutated. This recaps what we have just talked about in our notes. Here are a couple of questions to answer from your background reading, and then here's the actual gene. So this is normal myostatin. Here's the DNA sequence for the normal myostatin protein. You're going to turn it into messenger RNA, and then that's transcribe, and then you're going to translate. You're going to use the codon chart to see what amino acids would be strung together to make this myostatin protein. Then you're going to look at the Belgian blue myostatin gene. There's a mutation. So you're going to do the same thing for these guys. You're going to transcribe and translate, and you'll see what the mutation causes and sort of connect that back to, was it a point mutation or was it a frame shift mutation? Did it cause a significant effect or not on the overall protein that was being created? So you have a couple of questions to answer as a result of that. So do this, take your mutation notes, and then do this worksheet. Bring these back your Monday, Tuesday, first day of school that you're back next week, and we'll be ready to roll.